Hi and greetings to everyone. We are from Group 5 and our members include Rishi, FAIZ, RAHMAH, and I, JOCELYN. The social problem we choose to analyze and provide solution is plastic pollution in ocean. So here comes the topic of our presentation, plastic biodegradation to save the ocean. Looking at the content, we are going to go through the article, discuss about how plastic pollution in ocean occur, the impacts of plastic waste disposal, problem-solving methods that we will provide, and lastly, the possible implications of the results. The title of the article we found for our topic is More than 170 trillion plastic particles found in the ocean as pollution reaches unprecedented levels. The author of this article is Laura Paddison, and the article was published on the website of cable news network CNN on 8th of March 2023. The article has revealed some shocking statistics, estimating that there are around 171 trillion plastic particles in our oceans, weighing a staggering 2.3 million tons. A team of international scientists analyzed global data collected between 1979 and 2019 from nearly 12,000 sampling points in the Atlantic, Pacific and Indian Oceans and the Mediterranean Sea. They found a rapid and unprecedented increase in ocean plastic pollution since 2005. Disturbingly, without urgent policy interventions, the rate at which plastics enter our oceans could skyrocket by approximately 2.6 times between now and 2040. Plastic production has witnessed a significant surge in recent decades, especially single-use plastics. Unfortunately, waste management systems have failed to keep pace with this exponential growth. Only about 9% of global plastics are recycled each year. The problem is already visible in our oceans, air, soil, food, and even our bodies. We need concrete solutions, including reduced plastic production, sustainable alternatives, and comprehensive policies to combat this urgent issue. Now we are going to discuss on how plastic pollution in ocean occurs. Firstly, single-use plastic. The conveniences plastics offer, however, led to a throwaway culture. Today, single-use plastics account for 40% of the plastic produced every year. People are careless with plastic and leave it behind as litter after they use it for only once. Moreover, making plastic is inexpensive and the applications of the material are endless. Humans have invented plastic which is largely non-biodegradable, and plastics tend to persist in natural environment because of its properties. Many of these plastic products, such as plastic bags and food wrappers, have a lifespan of mere minutes to hours, yet they may persist in the environment for hundreds of years. Second reason is the laziness and irresponsible attitude of people. Plastics is so widely used by humans, it almost always ends up in the environment, causing more plastic pollution. Many lightweight single-use plastic products and packaging materials, which account for approximately 50% of all plastic produced, are not deposited in containers for subsequent removal to landfills, recycling centers, or incinerators. Instead, they are improperly disposed of at or near the location where they end their usefulness to the consumer. Studies from around the world have not shown any particular country or demographic group to be most responsible. The causes and effects of plastic pollution are truly worldwide. Next is the complexity in addressing plastic pollution. Addressing plastic pollution requires a combination of solutions that are complex, multi-sectoral, high cost, and country-specific. It requires putting a stop to leakage by improving solid waste management, building a more circular economy for public and private sector, and restoring ecosystems through clean-up. Last but not least, the pathways of plastic flow. One of the primary contributors to ocean plastic pollution is the substantial amount of plastic waste that finds its way into the seas. This waste originates primarily from land, as it is carried by rain, wind, overflowing storm drains, and the careless act of littering. Plastic carried by water flows downhill to the sea causes plastic pollution to reach the oceans. Since the ocean is downstream from nearly every terrestrial location, it is the receiving body for much of the plastic waste generated on land. Several million tons of debris end up in the world's ocean every year, and much of it is improperly discarded plastic litter.
The second one is the impact of plastic waste disposals. It will causes the increase of microplastic. Microplastic are tiny plastic particles that result from both commercial product development and the breakdown of large plastic. Marine species consume microplastic and introducing them into the food chain. They also pose a concern to the human health and the environment when they are built up in the tissue of fish or other seafood that human eats. From the article that we have here review, others have said that once a plastic get into the ocean, it does not compose, but instead it tends to break down into a tiny piece, and these particles are not really easily to be cleaned out, and we start with them. And the second one is causes a threat to the human marine life. Plastic trash in the ocean is harmful because marine animals can mistake it for food and get caught in it. Many marine animals such as sea turtle, seabird, dolphin, and whale are hurt or killed when they consume plastic or become entangled in the fish gear. Population decrease and biodiversity loss are the direct result of the health problem brought by a plastic, such as intestinal obstructions and the hunger. From the pie chart, we can see the percentage of marine animals that suffer from plastic dumping each year. When compared to other marine culture, the sea turtle is the most affected by plastic pollution, such as 35.1%. Why? Because floating because floating plastic trash is often mistaken by sea turtle for food. There is risk of suffocation, internal harm, and even death. The seabird is the second highest percentage animals. The seabird food chain is weakened because of human activities. Animals have a harder time to find food because of the abundance of floating garbage. As a result of having the tubular gland in the upper region of their stomachs damaged by a fibrotic disease, the majority of seabirds today resort to eating plastic in order to survive. And the third one is whale. Microplastic and hazardous compounds released from the plastic may be harmful for the whales, and the last one is seals. Unfortunately, seals frequently become trapped in the garbage that contain plastic. The seal will eventually starve to death as the plastic wrap gradually suffocate them. The third one is polluted by chemicals. Bisphenol A, or known as PPA, and phthalates are just two of many hazardous compounds that found in the plastic that can sweep into the environment and harm the aquatic organisms. Hormone disruptions, reproductive impairment, and long-term environment damage are all possible effects of chemical use, and it also causes an economic impact. Negative effects on the economy include the tourism, fishing, and coastal economic caused by the plastic pollution in the ocean. Communities who rely on the ocean for their livelihood and food securities are particularly vulnerable to the twin threat of marine pollution and biodiversity loss. And the last one, it will causes a global impact. About 70% of Earth's surface is covered by the ocean, and plastic trash can float 1,000 miles on the ocean currents. This means that the effect of improperly disposed plastic trash can be felt in the area that far apart from the primary source of contaminations. 40% of our garbage is from the bottle and under half of the collected for recycling, but only 70% become a new bottle. This statement means that they claim the claim that draw attention to the low rate of bottles recycling and the limited success of using recycled materials to create a new bottle. It points the necessity of enhance the recycling initiative and more effective process to boost recycling rates and decrease waste.
Enzyme as source of gene. In order to solve the plastic dumping problem, our group chose the scientific approach to solve the problem. The utilization of marine bacteria was inserted with the degrading plastic enzyme was conducted. So, to solve this problem, recombinant DNA technology was used in the lab and will be discussed by Rishi in the next slides. These plastics in the ocean mostly were composed of polyethylene terephthalate, PET, and also polyurethane. And these enzymes also are available to undergo DNA removal technology, such as catenase and lipase. From our research on several articles, we found out that chitinases and lipase were available to degrade this type of plastics. So we will continue with the definition of an enzyme. So we have to know the definition of an enzyme and also the mechanism behind the enzyme activity to justify why we choose gene from the plastic degrading enzyme to solve the plastic biodegradation problem. So an enzyme is a biological catalyst or protein which may speeds up the rate of reaction which occurs in the cell. The enzyme itself doesn't require very high amount of energy to produce the similar amount of products as if the reaction doesn't use enzyme also. So the mechanism of enzyme activity lies on the formation of enzyme substrate complex, which will break the substrate to yield the smaller products.
So the main goal of our group is to produce recombinant organisms that can degrade the plastics in the ocean. There have been many initiatives that have been taken to reduce the plastic waste from the ocean. However, it becomes harder year by year. The ocean is perhaps the most vulnerable environment to plastic waste. Once a plastic waste enters the sea, it has no boundaries. Waves and storms can carry plastics to even the furthest reaches of the ocean, where they accumulate into larger areas on the high seas or even become embedded in the shorelines and delicate coastal ecosystems. They have even been found on uninhabited islands. After some months or years at the sea, plastics break down into smaller and smaller pieces, battered by the waves and storms, eventually to sizes smaller than a grain of sand. This makes retrieving the plastics from the ocean extremely difficult, almost impossible. However, our recombinant organisms will be able to degrade those small plastic particles, including even degrading the big one. However, degrading the big one will take longer time compared to degrading the smaller particles. So before getting into the topics, let us know what is recombinant DNA technology. Recombinant DNA technology involves using enzymes and various laboratory techniques to manipulate and isolate DNA segments of interest. This method can be used to combine or splice DNA from different species or to create genes with new functions. The resulting copies are often referred to as recombinant DNA. One of the best examples is human insulin, because human insulin, which comprise a total of 51 amino acids, 21 amino acid alpha chain and 30 amino acid beta chain, was the first therapeutically used protein product resulting from recombinant DNA technology. So in our case, the genes of interest are chitinases and lipases encoding genes from the fungi such as Basidiomycota and also Ascomycota. So as FAIZ explained before, these enzymes are capable of degrading important component of plastics. We actually want to reduce plastics in the ocean. As we know, ocean has higher concentration of salt. Thus, we cannot directly grow fungi on the ocean, as higher salinity can kill the fungi, because as we know, the higher salinity or higher concentration of salt can denature the structural proteins of the fungi cells, including the membrane, the nucleus membrane, and all of that, due to the salting out effect. Hence, we chose a marine bacteria which is halophile, capable of living at higher salt concentration as the host. So we decided on cyanobacteria because it is the most common marine bacteria. Because cyanobacteria is previously known as blue-green algae, are the most primitive organism present on the ocean, Earth, in the ocean's Earth. They play a vital role as the primary sources of oxygen and nitrogen fixing agent in the aquatic environments. So now we are going to employ or going to use um, cyanobacteria as a host or carrier of these kind of genes that encoding for the plastic degrading enzymes. So now we are going to have a look on the methods of producing the recombinant organism, which is the cyanobacteria containing the gene of interest. In our project, the gene of interest is actually coming from the fungi, which is coding for the enzymes such as chitinases and lipases, which are going to degrade the plastics in the ocean. So before getting into the steps, I would say that this technology is quite expensive because we are going to use a costly enzyme such as the restriction enzyme. But the benefit or advantage of this step is we only need to perform this recombinant DNA technology only once at the initial stage of this project. Once we have done the rDNA technology, we then just copy the cyanobacteria as much as we want using the PCR method. So now we have the tools for our DNA technology. There are totally four. So let's start with the first one. The first one is the restriction enzyme. So restriction enzyme in recombinant DNA technology play an important role in deciding where the desired gene is inserted into the vector genome. Endonucleases and exonucleases are two examples of restriction enzymes. And then secondly, we got vectors. So vectors aid in the transport and integration of the desired gene into the host. These are the ultimate vehicles that transfer the desired gene into the host organism, hence they are crucial part of recombinant DNA technology toolkit. Because of their larger copy of number, plasmids are also known as bacteriophages, are the most commonly employed vectors in this technology. In our case, we are just going to use plasmids. So the vectors, are also known as plasmids, are made of the origin of replication, which is the nucleotide sequence where the replication begins, and then we get important gene markers such as antibiotic resistant, ampicillin resistant, and also cloning site. And also the cloning site is the site which will be recognized by the restriction enzyme. So, and then we also need the host organism. So in our case, the host organism is cyanobacteria. We chose cyanobacteria because it's one of the most common marine bacteria. And the last one is the organism containing gene of interest. In our case, is the fungi. So now we are going to focus on the methods. So now we are going to have a look on the steps of recombinant DNA technology. We have totally got 6 steps, but we are just going to look briefly on each step. So the first step is the DNA isolation. So our DNA coming from the fungi. So we need to extract or isolate the fungi DNA from the fungi itself. So in order to obtain the DNA, we need to make sure the DNA in its pure form without attached to any macromolecules. As we know, the fungi is eukaryote, so eukaryotes DNA will be attached to macromolecules such as glycoproteins or even the histone proteins in the form of nucleosome. So in obtain to achieve the pure form of the DNA, we need to use various enzymes such as lysozymes, cellulase, chitinase, ribonuclease, and proteases to degrade all those macromolecules that attach to the DNA of the fungi. After the DNA dilution isolation, we have obtained the pure form of the DNA. We are going to cut the DNA using the restriction enzyme digestion. So this enzyme will cut the gene at the designated place where we want to get the gene of interest only. Gene of interest in our case is the gene that encoding from the cutinases and lipases, which are the plastic degrading enzyme. So once we have cut the DNA, right, we are going to use agarose gel electrophoresis to separate only the gene of interest. So after we have done that, we are going to enter into the third step, which is amplifying of DNA. Now we are going to amplify or multiply the gene of interest containing the glutinase and lipase encoding. So for that, we are going to use PCR reaction. So once we get multiple copies of that smaller fragment, we are now going to put it into the plasmid. Plasmid is the carrier of the gene of interest. So for this, we need a fourth step, which is the joining of DNA using the ligase enzyme. So basically, we are going to attach the gene of interest containing the cutinase and lipase coding into the plasmid via ligation process. So after this, we will produce a new DNA molecule hybrid of containing the fungi of gene and also the plasmid of the bacteria. So because of this, these kind of DNA molecule are known as recombinant DNA molecule. So after this step, we are going to insert the recombinant DNA into the host. So in our case, the host is the cyanobacteria. So the cyanobacteria will receive this recombinant DNA molecules via a process known as transformation. Post-insertion, the recombinant DNA multiplies and manifests as manufactured protein under favorable condition. And the last step is the recombinant cell isolation. So This step is usually to screen or to filter out the cyanobacteria that have been successfully incorporated with the recombinant DNA molecule. So after all these six steps, we are ready with the cyanobacteria with the gene of interest. So now we can call the cyanobacteria as recombinant organism because it's containing the gene from other species, which is the fungi. So, but the thing is, the cyanobacteria will not express the enzyme casually or normally because in order to express the gene, it need to be exposed to the environment containing plastic. So for that, we need to breed or grow the cyanobacteria in the ocean so that it will be exposed to the smaller particles of the plastics in the ocean itself. So it will start to express the gene of interest, which is the cutinase and lipase encoding sequences. So now I would say we have successfully produced the cyanobacteria.
The implication of the result, it will produce a clean oceans. Restoring depleted or devastated marine ecosystem is another potential application of DNA recombinant technology. Changing the genetic makeup of a certain species may increase their efficiency in absorbing nutrients, promoting growth, and restoring polluted ecosystem. The goal of this technique is to improve the health and resilience of marine ecosystem by bringing them back into the state of equilibrium. Oil spill, plastic litters, and other harmful components are only some of the ocean contaminants that could be degraded by genetically modified microorganisms. The genetically modified organism (GMO) can generate enzyme or metabolic pathway that can degrade the toxin into harmless metabolite and making them easier to remove from the environment.
So one of the possible outcome of recombinant cyanobacteria is providing a healthy environment to the marine organisms because marine wildlife such as seabirds, whales, fish and also turtles mistake plastic waste for prey. Most then die of starvation or as their stomachs become filled with plastic. Because as we know, the main food of the turtles are jellyfish. So these turtles always mistake clear colored plastic as jellyfishes. As we know, our own digestion enzymes will not be able to degrade those plastics. So once the plastic is consumed by the animals, it will stuck forever into their body, into their digestion system. So literally, this will give a sensation of the feeling of full, so they don't feel hungry anymore although they don't receive any kind of nutrition or minerals or even the main component of energy such as carbohydrates, proteins and lipids from this kind of plastic consumption. So besides this, we would say that it is estimated that marine plastics are contributing to the death of more than 10,000 marine mammals every year. Plastic can affect marine species in varieties of ways, from entanglement and injury to ingestion and toxic contamination. The major determining factor in the size of plastics which can adversely affect different species in different ways and different timescale. So for example, large marine mammals are highly vulnerable to plastic entanglement, particularly from the ghost fishing gear. Fishing ropes, nets, that have been discarded or abandoned at the sea are often made from plastic and can trap and entangle a variety of marine life, from blue whales to small crabs. An estimated 300,000 whales, dolphins, and porpoises a year die from gauze gear entanglement. Marine megafauna are also vulnerable to plastic ingestion. In 2019, one whale was found washed up with 40 kg of plastics in its stomach, predominantly comprising plastic bags. So, apex predators are species that sit at the top of the food chain. A large yet understudied threat to marine apex predators, such as great white sharks and orcas, is the cumulative impact of microplastic in their food chain and bioaccumulation of toxic chemicals found in their plastic. A recent study has shown that a single plastic particle can absorb up to 1 million times more toxic chemicals than the water around it. Biomagnification occurs when chemicals build up in the fatty tissues of animals that have eaten or other contaminated species. The higher up the food chain you go, the greater the concentration of toxins. Orcas have been found with some highest deposits of chemicals in their fatty tissues and their breast milk, which is fed to their young. In this way, the contaminated toxics, it's even passed to their children also, the upcoming generation. Academics around the world are working hard to understand the link between microplastic pollution and also biomagnification of toxic chemicals. So we could say that plastic dumping is giving a huge negative impact on the health of the marine organisms and even poses a great threat to their existence itself. So, by implying or by using our recombinant cyanobacteria, there's a huge chance we could clear up the plastics, including the small particles up to the big plastics, so that we could provide a plastic-free environment to those cute marine organisms, so they could live happily in the ocean without consuming the toxic chemicals from the plastic. So, this is one of would be the greatest possible outcome, which is from the aspect of environment. So I would say, I would summarize that cyanobacteria, recombinant cyanobacteria, is actually an environment-friendly approach to overcome plastic dumping in the ocean.
Next, the recombinant DNA technology might bring social political benefits. Due to plastic pollution, many fish populations are becoming less productive, leaving fewer fish to support ecosystems and fishing opportunities. Many individuals, communities, and nations continue to rely on fish and other aquatic life as a source of food and raw materials. Therefore, recombinant DNA technology may reduce plastic pollution and build up healthy ocean. Healthy ocean guarantees there will be populations of ocean and freshwater wildlife for the future. With maintained fish stocks, fishers will have more stable income. In addition, by 2018, microplastics had been found in the organs of more than 114 aquatic species destined for our dinner plates, and evidence is rising on potential impacts on human health. So, recombinant DNA technology will clean the plastic in the ocean and protect aquatic species from plastic pollution, and thus promise safety food sources to humans. Consumers are more confident with the food safety and willing to buy.
The final implications of the result will be um, the biodegradation of the plastic in the ocean may save money for another expense. Based on the statistics from 2020 to 2021, between 4.8 and 12.7 million tons of plastic pieces are dumped into our oceans annually. And from that, 25 trillion macro and 51 trillion microplastics have been littered to our oceans. So, at over $90 billion, that cost includes programs to clean up the ocean trash, which in terms of clean ocean, for better manage the waste and improve the wastewater treatment plants. Um, it also means that investing research on biodegradable plastic. So, by while doing the biotechnological approach, it can save the money and reduce the use of money from for that field. It might reduce the expense of money. um, Therefore the money can be used to cover other economical goods, such as to reduce the deforestation at the forest and um, preserve our forests for the government objective. So besides, the money can be used to cover fishermen's economics and also cover the cover the expense for oil plantation. So I justify again that the last implication of the result is that biodegradation of plastic in the ocean can save money for another expense. Therefore, in conclusion, plastic dumping gives serious effects to humans, animals, and environment. Humans should never dump their plastics they use to the ocean. Plastic biodegradation, such as recombinant DNA technology, is a good technological approach and economically feasible to conduct. It is also environmentally friendly, as most available enzymes were used to extract the gene and good type of bacteria was used. Lastly, plastic biodegradation are important to reduce the plastic dumping and save the ocean ecosystems. We are Group 5 as RISHIGANESHAN as the leader of the group. Me, Muhammad Faiz, JOCELYN, Engkahyan, and RAHMAH HAYATI would like to say thank you, Doctor, for listening to our presentation. Saving the Earth starts with you.